Scancell is developing innovative immunotherapies for the treatment of cancer. So this field of immuno-oncology is a rapidly evolving field within the biopharmaceutical industry. There's probably not one major pharmaceutical company that's working in the field of oncology that isn't trying to build a significant franchise in immuno-oncology today. So Scancell has 2 platforms. From those platforms, we have 4 lead products, and we are addressing multiple cancer indications. So the 2 platform technologies we call ImmunoBody and ModiTope. And the immunotherapies that derive from those platforms essentially stimulate the immune system by presenting cancer antigens. So these are the unique molecular markers on the surface of cancer cells that discern them from normal cells. And that triggers potent T cell responses. And that's the T cells that are responsible for attacking the cancer. So within our ImmunoBody platform, we have 2 products, SCIB1 and SCIB2, and we'll hear a lot more about those in the future presentations today. And then in our ModiTope platform, we have, again, 2 products, Modi1 and Modi2. So what differentiates these 2 technologies? So before I go into that, let's just look at the cancer immunity cycle. So in cancer, the release of these cancer antigens are taken up by antigen-presenting cells or dendritic cells, which in turn prime and activate T cells, which then migrate and infiltrate the tumor where they can recognize, target, and kill the cancer. But in order for this to be effective at eradicating the cancer, it has to be a competent immune response. And that's what Scancell's technologies do. So in the case of ImmunoBody, it's the mechanism of action of how we actually present these antigens to these dendritic cells that induce these potent CD8 killer T cells. In the case of ModiTope, it's actually the antigens themselves. So Scancell is the first company to recognize the ability of citrulated epitopes or citrulated peptides to induce very potent CD4 killer T cells. And we have a strong patent real estate around that. And this has potential to be a game changer in the immunotherapy arena. But the net result is the same. So we're inducing 2 separate T cell responses, but the net result is the effective killing of cancer cells. So in our development pipeline, as I mentioned, 2 ImmunoBodies, 2 ModiTopes. ImmunoBody SCIB1 and the treatment of melanoma has finished a phase 1/2 clinical study, and we'll be hearing about the clinical data from that study from Professor Patel. And we're advancing that program into a phase 2 study in the United States, which we anticipate starting towards the end of this year. SCIB2 is for the treatment of non-small cell lung cancer, and we're advancing that program in collaboration with Cancer Research UK. And our ModiTope platform, Modi1, we expect to see enter the clinic in early half of 2019. And then Modi2 following thereafter, following additional preclinical evaluation throughout this year. And then another exciting development is this area of T-cell therapies through a collaboration with a company called BioNTech, which I will talk a little bit more about. So you might say at this point, Cliff, aren't there already immunotherapies in the market that are treating some of these cancers? And that's absolutely true. But there's significant opportunities for improvement on these existing therapies. You know, in terms of new cancer therapies to come in and replace standard of care or work in combination with existing therapies, but also to look at therapies that have improved safety profile, and that's particularly important when we're looking at combination therapies. We need to demonstrate efficacy in cancers where there are significant unmet needs. Not all patients respond to all therapies. Demonstrate durable responses so that we have that the cancer does not escape the therapy, and then looking at the delay and prevention of disease. In terms of how we might implement and take advantage of those opportunities, it's really trying to identify new targets like we're doing with our ModiTope platform, or new approaches to those known targets. Improve our understanding of the cancer immune cycle, to look at the synergies in terms of how we might implement our technologies with other targeted therapies, but other immunotherapies as well, and then focus on the hard-to-treat cancers, the significant opportunity there are a number of cancers that are just unresponsive to therapies. So just 2 recent announcements I would like to talk about is 2 collaborations that really give a significant endorsement and validation of Scancell's technologies. In December, we announced a collaboration partnership with Cancer Research UK around our SCIB2 product for the development in non-small cell lung cancer. And then shortly thereafter, we announced a collaboration with BioNTech in this area of T-cell receptor therapies. So I'll just talk a little bit about CRUK. As you know, this is a leading charity with a mandate to bring new and innovative therapies to cancer patients. And the collaboration that we have is that they've licensed our SCIB2 product, and they will undertake the responsibility for manufacturing and conducting a clinical trial, a phase 1/2 clinical trial in combination with a checkpoint inhibitor at their cost. So we look at this as a non-dilutive way to advance this program through the clinic. But we have an option to bring that program back into the company on the back of that clinical data. And obviously there's a financial option to do that, but it's a nominal fee. And if we do not undertake to take that option, then we enter into a revenue share agreement with CRUK if they decide to commercialize that independently. In the case of the BioNTech, this is a very different collaboration. BioNTech is Europe's largest private biotech company. They have collaborations with Genentech, Bayer, Sanofi, Eli Lilly, and just recently announced an A round of $270 million. And so they're obviously a very high-profile company. And one of the technologies they have is the development of these adoptive T-cell therapies, particularly around T-cell receptor therapies. And they have entered a research collaboration with Scancell to identify T-cell receptors that are directed towards the epitopes that we've identified for our MODI1 product. And at the end of that research collaboration, they have potential to take a commercial license to that technology and advance that forward. So we look forward to working with them and finishing that research collaboration and advancing this forward. But adoptive T-cell therapies have really started to become quite a huge amount of commercial interest in the industry over the recent months. Middle of last year, there was a $12 billion acquisition of Kite Pharma by Gilead, and then more recently we saw the acquisition of Juno by Celgene for about $9 billion. So you can see there's a huge amount of interest commercially within the big pharma companies to get access to these technologies. And through this collaboration with BioNTech, Scancell now has got a foothold in that landscape. So what are the near-term value drivers for the company? First and foremost, it's about getting clinical data, taking our SCIB1 product through the clinic and our MODI1 product through the clinic, and we anticipate seeing those clinical readouts within the next 2 years. It's about extending our pipeline through our research collaborations with BioNTech, and if we're successful with the CRUK Grand Challenge, again, extending our pipeline with a new cancer vaccine. Looking at technology partnerships like the one we announced with ICER Pharmaceuticals that help to de-risk our developments as we take these products forward. And then clinical partnerships with clinical network groups like CRUK, patient advocacy groups, but also key opinion leaders who are running our clinical trials. And I think this enables us to understand what is the utility of our products in a clinical setting. And then last but not least is industry partnerships. Identifying other companies in the industry, big pharmaceutical companies and biotechnology companies that have significant franchises in the immuno-oncology landscape and have products that we can see what are the optimum combinations of combining our products with theirs so that we can meet these unmet needs in patients.
I've been tasked with telling you about our new technology, which is called MODitope, or modified epitopes. And one of the challenges really when you're trying to induce an immune response against cancer is cancer cells look very like self. They look very like normal cells. And the immune system has been educated not to react and not to reject self. Otherwise, you're into autoimmune disease. Okay? But actually, it's not quite that black and white, because what the immune system is actually educated to look for is stress. So stress meaning a cell that's unhappy. Now that could be because it's infected, it could be because it's damaged, or it could just be because it doesn't have enough nutrients or oxygen, okay? And one of these processes called, autophagy, if a cell gets unhappy, if it's stressed in any way, what it starts doing is it starts digesting some of its cytoplasm. It puts it into a membrane vesicle, it will activate a variety of enzymes, and it will release that energy from those proteins, okay? One of the enzymes that's actually activated is called PAD, and what that does is it converts this amino acid in a protein, arginine, to citrulline. Okay? Now, if nothing else has happened, if there's no inflammation at that point, then that's exactly what happens. The protein will get digested and you'll get energy. But if at the same time as you've got stress, you have some degree of inflammation, it could be infection, it could be massive cell death, then you upregulate what we call this MHC molecule, MHC2, and that will present these citrullinated epitopes. And that's a red flag for the immune system. As soon as the immune system sees these citrullinated epitopes, it responds very robustly with these really exciting cytotoxic CD4 T cells. So the only thing that Scancell did that was particularly novel was recognize that tumor cells, despite what everybody thinks about them and the fact they grow and kill people, they're actually very stressed. They never have enough nutrients, they never have enough oxygen, and they have absolutely massive levels of this autophagy. Okay? So the question was, did they present citrullinated epitopes, and could we exploit them as novel targets? Clearly, I wouldn't be talking to you here today if I hadn't been right with that. And the answer to that is also, we have just got our European patent basically granted, and that's for the use of any citrullinated peptide for the treatment of cancer. Again, underpinning that we were the first group to realise the potential in cancer. So what we've done is we've screened a whole variety of peptides, we've identified a whole number of epitopes, 3 that we're particularly excited about, 2 from a protein called vimentin, which is a common cytoskeletal protein, and 1 from enolase, which is a common glycolytic enzyme massively overexpressed in cancers. Now, these particular proteins are expressed very highly in triple-negative breast cancer. So triple-negative breast cancer is a cancer that does not respond to hormone or other types of therapy, so it's a very acute disease. Ovarian cancer, which does respond to chemo, but if you fail the chemo, unfortunately, it has a very dire prognosis. And osteosarcoma, okay, which again, if you don't respond to chemo, has a poor prognosis. And what we've basically shown is a combination of those 3 peptides in a very aggressive tumour model gave us 100% survival. In other words, it rejected every single tumour in every single animal. Okay. I have never in 30 years seen anything that potent with any of these vaccines. These cytotoxic CD4 cells are really amazing. But we haven't just sat with Modi-1. Modi-1's now moving into manufacture and it will go in the clinic. But there are many citrullinated proteins. There are over 37 that have been recognised to date. Okay? We've looked at 7 and all 7 have given anti-tumour responses. So we still have another 30 to go along the way. They may or may not all give anti-tumour responses, but so far it's looking good. So the CRUK have decided that to have a non-toxic effective vaccine would be a huge step forward in cancer. Okay? And in order to do that, they have given this 20 million basically as a grand challenge to say who can come up with an effective cancer vaccine. Okay? It really needs to be done in this field of immuno-oncology. Okay? Now, because we were already working with BioNTech and indeed ICER in terms of our adjuvant and our collaborations, and BioNTech worked very closely with Genentech, we all got together and decided that we thought we could answer this grand challenge. So in essence, what BioNTech and Genentech do is they'll take your tumour, they'll sequence it, they'll identify all the mutations within your individual tumour, and if any of them will stimulate a T-cell response, they make an RNA vaccine and they basically immunise. And that's a really interesting technology. It's already proven to work in some melanoma patients. But only about 10 to 20% of patients will have enough mutations for this vaccine to work. So both BioNTech and Genentech are very excited about the potential of Moditope. Because as I said to you, Moditope is not just for one patient. Indeed, it's not just for one cancer. It can work in a whole range of different cancers. But clearly, at this stage, it's very novel. We need to get in the clinic. We need to prove it works. So the grand challenge is about an older technology that's working, but only in a subset of patients, and a very exciting technology, Modi-3, from Scancell, that potentially could be a game changer in terms of this field. So we're going to develop this Modi-3 basically, and that's against the cancers we've chosen to go in the grand challenge are head and neck cancer, glioblastoma, lung, and pancreatic cancer. Potentially because they have one of the worst survival rates for any cancers. Okay, so we've taken that on the nose and we're going to try and go with these really difficult cancers with these 2 approaches. So for Scancell's perspective, not only does it get their technology exposed to key opinion leaders, because we have over 21 of the best scientists in Europe and the US on this grand challenge, so we get huge exposure, but we also take one of our lead products, Modi-3, into the clinic in a different cohort of patients. Again, Cliff's already mentioned BioNTech. So BioNTech, as you say, one of the largest private companies. They have currently over 150 staff, all focused on immuno-oncology, so really exciting. So basically, Professor Tahin, who's head of BioNTech, is extremely interested in Moditope. Okay? He's really intrigued by the whole concept. But what he wanted to do initially was just identify the T cell receptors on these CD4 T cells that were stimulating this response and see if he could identify them and use them for adoptive T cell transfer.
My role today is to tell you about let me advance this first so you can see what I'm talking about. The ImmunoBody platform, which is Scancell's other platform. Professor Darren's told you about Moditope. The ImmunoBody platform is the other main platform that they are developing. So when you're looking at new immunotherapies, I think one important thing to ask yourself is how is this different from what everyone else is doing? And there is, as you know, as analysts in this field and professionals looking at this field, there's so much work going on at the moment. I think one of the key things that Professor Durrant and her colleagues at Nottingham have been able to do is to show that a key element of any immunotherapy has to be developing high avidity responses. So it's a high avidity T cell responses, and that's what the ImmunoBody platform allows them to do. And I'll show you why that seems to be the case. But it's only the high, and avidity is really a measure of the interaction between the T cell and its target. And it's only those high avidity cytotoxic T lymphocytes that are able to be selected into the memory pool and to mediate tumor eradication. And the ImmunoBody platform allows people to do that, allows you to do that with CD4 and CD8 T cells. So let me show you a little bit about what that looks like. So, like everything that Scancell's doing, it's well protected by a patent, by patents for the whole platform. The T cells, the T cell epitopes can be, you can pick any T cell epitopes that you want to that target tumors. You'll see later on that Professor Patel will show you the data on the first program, which was the SCIB1 program. I'm going to talk to you about the SCIB2 program. The ImmunoBody is delivered to the tumors or delivered to the patient as a DNA plasmid that has the instructions for producing this, what we've called here a genetic antigen antibody complex. So you have the epitopes inserted into the CDR regions of the ImmunoBody. At the moment, that's delivered through a process of electroporation. There's also work underway to look at a nanovesicle delivery of that, which would be slightly easier for the patients. And as I mentioned, there's the SCIB1 program for melanoma and the SCIB2 program for lung cancer. So SCIB1, we've completed a phase 1/2 trial, planning a phase 2, and it's a SCIB2 that's in partnership with the CRUK. But I want to tell you a little bit about why this is different from other vaccine technologies. So the way this DNA plasmid is taken up into cells, there are two different pathways. So there's a direct pathway where the plasmid is taken up by the antigen-presenting cell itself, produces the antibody, and that is processed then to present to the T cells. The indirect pathway, or the cross-presentation, is through this taking up of the DNA of the ImmunoBody by another cell, in this case a smooth muscle cell. That then produces and secretes the antibody, and then that antibody is also taken up by antigen-presenting cells. And the work that's been going on in Nottingham over all these years, it has been some time, through deleting various parts of this pathway or modifying it, Professor Durrant has been able to show that it's only when you have this cross-presentation that you get the high avidity T cell responses, and it's only when you get those that you get the effective anti-tumor response. So we picked lung cancer for the reason that lung cancer is still relatively poorly treated despite the advances that have occurred in recent years. Lung cancer divides up into two main types, non-small cell lung and small cell lung, with non-small cell lung accounting for the majority of the cancers. It accounts for over a fifth of deaths from cancer in the UK. And so it's not only a common cancer, it's also a very lethal cancer. And recently we've seen success with checkpoint inhibitors treating this and beginning to displace chemotherapy as the main form of treatment for non-small cell lung cancer. But when you dig into the numbers and understand who's really benefiting from that, only if you take the first-line therapy as a monotherapy for Keytruda, which is the main checkpoint inhibitor, one of the main checkpoint inhibitors that's been developed for lung cancer, only 30% of patients are suitable for monotherapy by pembrolizumab. They have to have a high PD-L1 expression, so that's the counter target for PD-1. And only 45% of those patients will respond. Still a high response rate in that selected group, but 45% of 30% is only 15% of patients who are benefiting. And when you look at the second line treatment, the numbers are even smaller. And when you look at some of the benefits in second line, the benefits are quite marginal. With the other checkpoint inhibitor, nivolumab, the gain in median progression-free survival is a matter of 3 weeks. So you can see that there's a huge need to, immunotherapy clearly has a potential in the treatment of lung cancer, but somehow we're not quite recognizing that full potential yet. So as Cliff mentioned, this is the subject, SCIB2 is now the subject of a collaboration with Cancer Research UK. And it's important to recognize, I think, that when we think about Cancer Research UK, they are a research charity. They're not just a charity thinking about giving money to cancer and to cancer patients. They are a research charity. They formed from the premier research institutions in the UK. And I think this is very much a validation, again, of Scancell's work. These are scientists who are only taking on programs. I know from my own background working in companies in the States, we've had collaborations with Cancer Research UK. You only get in the door if you've got something they think is interesting and valid. And they've obviously been very interested in the work that Professor Durrant and her colleagues have done, form this collaboration. This allows Scancell, as Cliff mentioned, to effectively conduct a program without having to put the investment into that, allowing them to free up their own money to do other programs. They're not short of ideas here, so there's not like the money's not going to be used for other reasons. It allows Cancer Research UK to do the program and then having the option to buy back when you are aware of the data, once the data is finalized.
I'm a clinician and a scientist, so half my job is seeing patients with cancer, trying to develop and test novel therapies. And I've got to say, with immuno-oncology, with the advent of immuno-oncology, this is the most exciting time to be an oncologist, a time I'd never thought 10 years ago that we'd be seeing. And that's because we're beginning to see developments which are making really big changes in my everyday clinic. There are about 2,500 deaths from melanoma. Thankfully, most patients are cured because you see a mole, you can cut it out, and you can cure it. However, those 2,500, although you've cut out the primary and you thought you might be cured, you're not. And that's because in melanoma, what happens is the cancer cells which develop in the moles of your skin, is there a pointer in that? Yep. Start invading downwards and then can invade the blood vessels or the lymph vessels and spread. And unfortunately, if your melanoma is quite thin, 1 millimeter thick, so thankfully for those people, you've got a 94% chance of being cured. However, if your melanoma is 4 or 5 millimeters thick into your skin, then cancer's already spread, and your chance of being alive at 10 years is less than 40%. And that's because melanoma can spread either in the lymphatics, and that's called stage 3 melanoma, so it's already spread, but in a proportion of people, about a third, you can still cure it with surgery. But once it's gone through to stage 4, which is lungs, brain, liver, or any of these organs, the chance of cure, unfortunately, is really bad. I showed you a slide earlier on where you have where the cancer makes things which stops dendritic cells working. And with SCIB1, you can vaccinate away from the site of the tumor, switch on the immune system so that when the brakes are taken off, you can work even better. What we did, I won't talk you through how SCIB1 works, but you've heard about that. What we did was we took that vaccine and took it into patients. We did a phase 1 trial, first in man, across several main centres in the UK, patients with advanced cancer, which had either still had their cancer in situ, which we could see, or had their tumours resected. So there were 15 patients with tumour present and 20, and they received vaccines along the way. And the three main things I think to take away from that phase 1 trial was, the first bit was it's safe to give. The toxicity profile was really good. That tells me that it's a really focused immune response. The second, which I wasn't sure we would see with just a vaccine on its own, was that we actually saw tumour regression. So we saw lumps of cancer, and that's one here, patient with lung metastases. There, that's about a centimetre and a half lump, which you can see on a CT scan. That bit there is down and regressed. So that was telling me that not only was it setting off an immune response, but enough to cause a cancer shrinkage. And then the bit which I think is really important is in that proportion of people who had had their cancer taken off, we knew would have had microscopic deposits elsewhere, because we know from historical data that those patients do, those types of patients, that actually the 5-year survival rate was out here. This is not just survival, these people who haven't recurred, but 14 of the 16 were all fine.
Just to wrap up a little bit, about 20 years ago, there was a venture capitalist from Kleiner Perkins in Silicon Valley who made an odd kind of statement, an audacious statement. And he basically said, his name was John Doerr, and he basically said, I think it's quite possible the internet is underhyped. And why that was so audacious at the time, 20 years ago, the most overhyped thing on the planet was the internet. So you were thinking, what sort of world is he seeing to say that it's underhyped? But of course, he was correct. So over the last 20 years, the growth of internet companies, thousands of them, even what they call the FANG companies, Facebook, Amazon, Netflix, and Google have enjoyed phenomenal growth over that time. So perhaps it's an odd way to wrap up. So you might think, what's this got to do with Scancell or immuno-oncology? Well, the fact is, it's got everything to do with Scancell and immuno-oncology. So I'm here today to basically take a leaf out of John Doerr's book and say, immuno-oncology is probably underhyped. And to Professor Patel's comment, we've only really started making progress in this field. It's been a long road. There was a physician practicing in New York about 100 years ago called William Coley, who noticed that certain patients that had certain diseases, their cancer improved. So he began working on something called Coley's toxins, Coley's vaccines, they became known as, to explore the complex relationship between the immune system and cancer. And now we're finally making some substantial progress. So in 20 years, maybe we'll know, but there's tremendous progress still to be made in immuno-oncology. And I'm just delighted that Scancell is absolutely at the forefront of that.